Mountain of Roses, everyone. So my topic is all about genetic technology. So genetic technology or engineering, DNA technology, is a process of making changes in the DNA code of living organisms. Uses DNA technology to cure diseases, treat genetic disorders, improve food crops, and etc. How do we make changes to DNA of an organism? Using restriction enzymes. Bacterial enzymes act like scissors that cut DNA molecules at specific sites, create stick ends that can easily bind to complementary DNA and form new sequences of nucleotides. Plasmids are ring of DNA found in a bacterium, which bacteria, in addition to its main chromosome. These plasmids can be cut with restriction enzymes, allowing other genes or a donor genes to be spliced into the plasmids. Once gene has been spliced into plasmids, the plasmids is returned to the bacteria. Plasmids are the bacteria then divides, giving copies of the new gene. Recombinant DNA, combination of DNA from two or more sources. Example, inserting human gene for insulin into bacteria in order to produce large amounts of insulin for people whose bodies do not produce enough. And this is the illustration of insulin. More DNA technology techniques. DNA fingerprinting. Pattern of bands made up of specific fragments from an organism's DNA. Uses determining relationship between individuals, determining how closely species are related, helping solve crimes. DNA fingerprinting, how it works. Use RFLP analysis to omit, extract DNA and cut it using structured enzymes. Gel electrophoresis. DNA placed into wells made on a gel. Fragments or segments of DNA are then separated based on size and charge. And this is the illustration then. Next is the gel electrophoresis animation. Polymerase chain reaction of PCR, used to quickly make many copies of selected segments of DNA, only requires tiny amounts of DNA. Human Genome Project began in 1990. So to determine the nucleotide sequence of the entire human genome, approx 3 billion nucleotide pairs to map the location of every gene on each chromosome, improve diagnosis treatments, and cures for genetic diseases. Cloning. A group of genetically identical cells maybe produces asexually or mitosis is a growing body parts. Gene therapy. Treating a genetic disorder by introducing a gene into a cell or by correcting a gene defect in a cell's genome. Involves ethical decision. Example, insurance companies. Practical uses of DNA technology produce effective vaccines. Interesting research, scientists are creating a banana vaccine to vaccinate children against viral diseases like hepatitis B. Practical uses of DNA technology increases agricultural yields by protecting plants by transferring genes that are harmful to pests into the plants, producing plants that are resistant to weed controlling chemicals, herbicides, developing plants that do not require fertilizers to grow. Safety and environmental issues. Genetically engineered foods may contain toxins, making people allergic to the foods. Genetically engineered crops may take over the native plant species,